cells of the immune system, such as macrophages and dendritic cells, are the first line of defense in recognizing pathogens of various kinds. These cells have developed several kinds of receptors for recognizing different kinds of pathogen-associated molecular patterns, known as PAMPs. There are different classes of these proteins that recognize different types of PAMPs. Toll-like receptors, or TLRs, are composed of multiple leucine-rich repeats that are useful for recognizing various PAMPs. TLRs are membrane-associated proteins. Some are located on the surface of the cell, while others are located on endocytic vesicles, where they survey the degraded contents of pathogens taken up by endocytosis. Each member of the TLR family recognizes different kinds of PAMPs. For example, TLR5 recognizes flagellin, which is a highly conserved constituent of the bacterial flagellum. Bacterial genomes contain methylated CpG oligonucleotide motifs, which are recognized by TLR9 once the genome has been degraded in the lysosome. TLR6 and TLR2 are a dimer that recognize diacyl lipopeptides. TLR1 and TLR2 are a dimer that recognize triacyl lipopeptides, and TLR4 recognizes lipopolysaccharide, LPS, a component of gram-negative bacteria. Like TLR9, TLR3 and TLR7 are located on endocytic vesicles and recognize double-stranded RNA and single-stranded RNA, respectively. When any TLR is activated, it sends a signal to the nucleus by activating transcription factors. Not all pathogens, however, live in the extracellular space or are phagocytosed. Some pathogens, such as viruses, exist and replicate in the cytosol. There are at least two classes of receptors that can detect pathogens in the cytosol and signal their presence to the immune system. One class of such receptors are members of the nucleotide oligomerization domain family, or NOD proteins. For example, the NOD2 protein, which is located in the cytosol, can detect bacterial proteoglycans of intracellular bacteria. When the NOD2 protein recognizes its ligand, the muramyl dipeptide, it sends a signal to the nucleus to activate transcription. Finally, there is a class of intracellular receptor proteins that contain an RNA helicase domain and two caspase recruitment domains, or CARD domains. One member of this family, RIG-I, recognizes double-stranded RNAs that are a component in the life cycle of many RNA viruses. This class of proteins also sends a signal to the nucleus, but unlike TLRs and NODs, it activates the production of type 1 interferons. In all, the toll-like receptors, NOD proteins, and the RNA helicase CARD domain family provide the innate immune system with the ability to detect both extracellular and intracellular pathogens and to activate an immune response against them.